About the subdivisions of phylum Porifera. So far we have discussed the general characteristics. So let us begin our lecture with the subdivisions. So you draw uh, three boxes and at the top you write phylum Porifera. So phylum Porifera can be divided into three classes. And what is basis of division? Basis of division is again the skeletal element. Remember, underline it. It is a skeletal element, means the composition of skeletal element in the animal. Okay. And second is the distribution or the localization of one very important cell, that is collar cell. You remember, collar cells or coenocytes are unique to the poriferans. Now you put a line like this. So the phylum Porifera can be divided into three classes. First is called Calcarea. First is called Calcarea or Calciospongia, and second is called Hexaactinellida or Hyalospongia, and third is called Desmo Spongia. So if you recall the skeletal element, so basically it can be divided into two things. Yes, please, Ashish. Yes, he is saying the spicules and spongine fibers. Those are made of proteins. But so the basic element is, if the spicules are composed of calcium carbonate, then what we call it, where we will place the poriferan? In calcarea or calciospongia. The name itself indicates the what type of skeletal element it has. Second is Hexaactinellida. Here the spicules, again here in calcarea and Hexaactinellida there are spicules. So in Hexaactinellida there are siliceous spicule composed of silica. Now come to the third one, that is third class, called Desmo Spongy. So Desmo Spongy actually composed of either siliceous spicule or spongine fiber or both. But how to differentiate between Hexaactinellida and Desmo Spongy? How we can differentiate? Because both have the siliceous spicule. So it can be easily differentiated because in Hexaactinellida there are tri-radiated spicule or hexaster. So where we are using a term called axon here. Axon is actually part of neuron, are the spines that is breaking out of the neuron. So here we are using this term for the pointed structure in spicules. So let us see the characteristics of each one of them. The first is the skeleton of calcarea or calciospongia. Here the spicules are, there is no doubt, it is composed of calcium carbonate, but they are either monoradiated, triradiated, or tetraradiated. As we can see here, there are 2-pointed projection, there are 3-pointed projection, and there are 4-pointed projections. Now come to the Hexaactinida. Hexaactinida, there are triradiated spicules. But if two triradiated spicules, they fuse together, there are 6 rays coming out. So that's why the name Hexaactinida. But remember, here the spicules are siliceous. Okay? And always there is presence of hexaster spicules, means spicules with 6 axons or 6 projections. Now come to Desmo Spongy. In Desmo Spongy, spicules are obviously they are siliceous, but they are never triaxon. They are never triaxon. कभी भी spicules triaxon नहीं होगा. Now, let us see another thing that is important in Desmo Spongy. Desmo itself means the ligament, the connections. Means there are something that connects thing. That is here spongin. Spongin fiber, that are protein fibers. So what we can see here, that in calcarea there are calcareous spicule, in Hexaactinida Specules 6-rayed spicule, in Desmo Spongy there are siliceous spicule, but they are not triradiated and with spongin fiber. Now come to the distribution of the Coenocytes. So on the basis of distribution of Coenocytes, there are different type of canal systems present. When we discussed about the Porifera general characteristics, we have discussed the type of body organization on the basis of the canal system. Here we have shown 3 kind of canal system. The Ascon type, the most simplest type, Sycon type is mediocre, and Leucon type is most complex type of canal system. So in calcarea, all the 3 type of canal system is present. But in case of Hexaactinida, there is only Syconoid and Leuconoid type of canal system is present. There is no Asconoid. Now come to Desmo Spongy. In case of Desmo Spongy, there is no Asconoid, there is no Syconoid, only Leuconoid type is present. Means there is a most complex type of canal system in Desmo Spongy. Now let us see the distribution of Coenocytes. How many of you remember that Coenocytes are the most peculiar cells in Poriferans? And collar cells, they are also called as collar cells. And when we discuss about the general characteristics, the Poriferans, they have a cavity enclosed. That cavity is called paragastric cavity or spongocoel. So in case of calcarea, the entire spongocoel is lined by coenocytes. So means the coenocytes are confined in the paragastric cavity or the spongocoel. But in case of Hexaactinida, they are confined in the finger-shaped chamber. The finger-shaped chambers, they are confined. The shape is very important here. The chamber shape is very important. But in Desmo Spongy, In Desmo Spongy, they are confined in a round-shaped chamber. They are confined in a round-shaped chamber. Remember, if you ask me most beautiful, means they are beautiful in look and their structure is so transparent, so glassy. That's why the name hylo-spongy. And why they are so glassy structure? Why they appear glassy? Because there is no exopinacoderm. Remember, in case of Hexaactinida, there is no pinacoderm. There is no exopinacoderm. There is no exopinacoderm. Exopinacoderm is not there. And one more important thing, that all the poriferan classes all the poriferans, they are marine, but there are few, those are freshwater, and one of those exceptions that belongs to Desmo Spongy. Remember, the Calcarea and Hexaactinida, they are not freshwater, they are always marine. But the Desmo Spongy, there are some family called Spongilidae that we will discuss later also, that is freshwater sponge. Okay, it is often asked in the examination that which of the following is freshwater sponge. Okay, it may be asked on the basis of class or on the basis of animal. Now, come to the further classification of classes. So now we have 3 classes. So we will see one by one. Okay. Now we have a further systematic classification within the class, that is the orders. The first class was Calcarea or Calciospongy. Here skeleton is made of calcium carbonate. And the distribution of Quinocyte is very important here. On the basis of distribution of Quinocytes are collar cells. You remember collar cells? I know. Okay. So the distribution of collar cell is very important. On the basis of that, there are 2 orders. There are 2 orders. One is called Homocoela. Coela here is which cavity? What kind of cavity is there in poriferans? Yes, please. Yes, Assis. Okay, he said that it is spongocoel, are also called as paragastric cavity. This cavity is entirely lined by Quinocytes. Then the animal or the calcareous sponges belongs to Homocoela. And if their distribution is not uniform in spongocoel, then it is called heterocoela. Okay. Now come to the further characteristics. So why this distribution is heterogeneous? Because the body wall is unfolded. Body wall is unfolded. We have talked about the body wall. There are 3. The pinacoderm, the mesenchyme, and then the quinoderm. That is body wall. It is unfolded structure in case of Homocoela, but it is folded structure in case of Heterocoela. Now come to the distribution of Quinocytes. The Quinocyte line the spongocoel cavity. Entire spongocoel cavity is lined by Quinocytes. But in case of Heterocoela, since the body wall is folded, so there are formation of canals. One is called incurrent canal, another is called radial canal. So a radial canal is lined by coenocyte. That's why that is called flagellated chamber. Flagellated chamber. Okay. Now come to the type of canal system. Here the body wall is not folded. So there is most simple type of canal system, that is asconoid, and the shape of animal is either cylindrical or vase shaped body. But in case of the heterocoela, the syconoid or leucon type of canal system is present, and body shape is again vase-like. That's why we have the example, the leucosolenia. Leukos means white tubes. White tube-like structures are there in Homocela. Example is leucoselenia. It is often asked that what type of canal system is present in leucosolenia. So will you take the leucon type? No, there is a scon type of canal system. Now come to heterocela. There are two examples. One is we will discuss in detail, that is Skyphar sycon. There is syconoid type of canal system present. Now this is over, and we will discuss the next class, that is Hexaactinellida. In Hexaactinellida, here shape of the axon projection terminal, the shape of axon projection terminal is very important while classifying the Hexaactinellida. So Hexaactinellida can be divided into two orders on the basis of the tip of axons, on the basis of the tip of axons. The animals are like cage. They form like a cage-like structure. They form a cage-like structures. Okay, so they form a cage-like structure. And here there are two orders, Hexasterophora and Amphidiscophora. Remember, in case of Hexasterophora, the specules are, yes please, hexaster. So there are 6 rays, but here at the tip there are bifurcations, means axis is divided into 2. Okay, like this. And like this. So this is Hexasterophora. Okay, but in case of Amphidiscophora, what will happen? There are 6 rays specule again, because this 2 orders belongs to class Hexaactinellida. But here at the end there are disc-like structure like this. Okay, and so there are disc-like structure like this. That's why the name amphi means both the ends of specules with disc. So there are 6 disc. So ends there are disc-like structure, and it is curved inside at the center of the axis. Now let us see other characters. So now it is clear to you why those names are given. So hexaster spicules with branched axis at the tip or at the end. But in case of Amphidiscospora, spicule are amphidiscus. They are amphidiscus and convex disc at the end. Here the axis is branched. Okay. Now come to the flagellated chamber. Whenever I say flagellated chamber, the only flagellated cell that is present in poriferans is coenocytes or collar cells. So here in case of hexasterophora, suppose this is the body and we have taken the cross section. So these flagellated chambers, they are regularly and radially arranged. So they are radially arranged like this. Suppose these are the chambers. Okay. So here the flagellated chambers are regularly and radially arranged. But in case of Amphidiscophora, there is unusual shape. They are not arranged regularly. Okay. Now come to the attachment. If you see the structure of the poriferans, suppose this is a poriferan that I am drawing here. So it is attached with the substratum. Okay, suppose this is the substratum. But in case of Amphidiscospora, there is shape like this, but they have lot of projections that are coming out. Lot of fibrous projections that are coming out. These are appearing like root. Okay, these are appearing like root. Okay, and that's why they have unusual structure, that is root tuft is present. Where they are attached with the substratum, there is root tuft. So root tuft is present here, but it is absent in case of hexasterophora. There is one very important example that you always remember, that is Euplectella. Why I'm giving so much time on Euplectella? Because it is often asked in the examination. Venus flower basket. Venus flower basket. It is a gift that is given in Japan. Why it is given? Because the Euplectella, if you see here, it is a cage-like structure. Okay. So somebody is there in the cage, it cannot escape. So what happens during the development of Euplectella? There are two shrimps that occupy this cavity of the Euplectella and remain there. If the Euplectella die, then only they will die or they will separate. That's why for this kind of union, they present Euplectella as a gift for newly married couple. So this story will keep you remember this example, Euplectella. Okay. Now come to the Amphidiscospora. There is Hylonemma, that is called glass rope sponge. What we call it? Glass. If you see the animal, there are so many rope like structure. And why the name Hylonemma? Because there is no exopinacoderm. So they appear like a glass. Glassy fibers are coming out. Okay. Now come to the third class, that is Desmo Spongy. So in Desmo Spongy, we have subclasses. Okay, in first two classes we do not have subclasses, but here the animal structure is different. That's why there are three subclasses. It's based on whether, again, the skeletal component is very important. If spongine fiber is present or not, the spicules are present or not, what type of spicule you would expect in case of Desmo Spongy? Yes, please. Asutos. Calcareous. He is saying calcareous. No. Make a big cross on it. In Desmo Spongy, spicules are siliceous. Okay. Now, see. There are first class, that is Tetra Actinida. So tetra means 4 rays. Spicules are 4 rays. Okay. Monoxonida. Spicule has 1 rays. 1 rays spicules are there. And now we come to Keratosa. In Keratosa, there is no spicule at all. ये अंडरलाइन करके आपको लिख लेना है. In Tetra Actinida, there is no spongine fiber. In Keratosa, there is no spicules. Okay, so they are contrast. Tetra Actinida and Keratosa are having contrasting feature. Okay, let us see. In Tetra Actinida, tetraaxonoid spicules are either present or absent. In Monoxonida, the monoaxon spicules present or absent, but with or without spongine fiber. So it is intermediate somewhere. In Keratosa, there is no spicule. There is no spicule. Remember, only spongine fibers are present. That's why we have a very good example that we use in our toilets. That's why we have example here in Keratosa, that is toilet sponge, that we will discuss. Now come to Tetra Actinida. So in Tetra Actinida, there are 3 orders: Myxospongida, Carnosa and Corystida. Remember, in Myxospongida, there is no spicule at all. There is no spicule at all. In Carnosa, spicules are small in number. Spicules are small in size. Only small size spicules are present. In Corystida, only large size spicules. Large size spicule as well as a small size of spicules are present. So what we can see, in Myxospongida there is no spicule. In Carnosa there is equal size spicule and there is small also. And in Corystida we have large spicule and small spicule also, but remember they are tetraaxon type. In Myxospongida we have Oscarella and Helicerca as an example, and Planchina is very important. It is often asked in the examination. So Planchina is here, belongs to order Carnosa. So here equal size spicules are present. And Corystida, Thania is very important. It is often asked. Both small and large size of spicules are present. Now, come to the Monoxonida. In Monoxonida, there are 4 orders. First order is called Hydromerina. Second is Helicondrina. Third is Poecilosclerina. Then fourth is Haplosclerina. So what is the characteristics? So see here. In case of Hadromerina, there are spicules, okay, and they are dumbbell shaped, means amphidiscus spicules. They are monoaxon. See here, they are monoaxon. They are monoaxon. They are monoaxon, but they are having disc on either side. So that's why amphidiscus spicules are there. And star shape, small spicules are there. But remember, there is no spongine fiber. There is no spongine fiber. And here there is very good example that often asked in the examination, that is Cleona. This is very important. When I say important, means it's very important. Why it is important? Because Cleona is also known as boring sponge. They create hole. They create pores. The ships that are made of wood. In wood, they are parasite and they create holes. That is good for ship or a boat or not. So that is bad for boat industry. Okay. So Cleona, you remember, that is a boring sponge. Okay. And it has no spongine fiber with amphidiscus spicules. In Helicondria, Helicondria is very important. How many of you enjoyed the rusk and tea? That is dry bread and tea. So if you recall the texture of the rusk or the dried bread that is given to you, that is same as the example of Helicondrina. That's why they are called as crumb of bread sponges. They are called as crumb of bread sponges. But here you see the spicules are small as well as long with few spongine fiber. Remember, there are very small amount of or very little amount of spongine fiber. Okay, but it is present. And here is small as well as long. Now come to Poecilosclerina. It is very important because it is often asked that where there are sickle shaped spicules are present. So it is present in Microceona. So this example is very important. It is also often asked in the examination. Here you see the spicules are sickle shaped. Okay, they are sickle shaped and they entangled with the spongine fiber. There are a lot of spongine fiber. In Haplosclerina, there are long axonoid spicules. There are long axonoid spicules. I have not made here the spongine fiber like this. There are so many spongine fiber in this also. Okay, so there are so many spongine fiber. So spongine fiber is there and only long spicules are there. And here one very important example again, that is spongilla. Remember, freshwater sponge, that is called spongilla. Spongilla is freshwater sponge. Spongilla is freshwater sponge. Okay. Now come to the two other example, that is Kalina. Kalina is what? There are finger-like projection, like somebody is projecting his hand out of sand. That is Kalina. Okay. Then there is Haliclona. Haliclona is finger-like projections. Okay. So they appear like a small tree in the seawater. But Spongilla is only freshwater sponge. Remember. Now come to the last one, that is Keratosa. It is very important because it is a horny sponge. There are a lot of projection coming out of the sponge and they are very dull in their color. Often they are brown and they appear like leather. They appear like leather. And here we have very good example. That is Euspongia or Spongia. Remember, people often confuse with Spongia and Spongilla. Remember, Spongia is without any spicule. That's why this is called toilet sponge. We often use it to remove dead skin. Okay. And second example is Phyllospongida. Phyllo means leaf-like, and Hippospongia, horse-like sponges. So these are the very good example of poriferans. So you always remember the Cleona, Microceona, Spongilla, Spongia. Okay. So I hope you all understand the basis of classification and what is the basis as well as how to classify animal. Suppose I take one example, that is Spongilla. Suppose I ask somebody to classify Spongilla. So how to classify? First we will write the phylum. Then phylum is Porifera. In phylum Porifera we will write certain characters are there that we will write. Then we will come to class, that is Desmo Spongy. Obviously there are siliceous spicule but not tri-radiated with spongine fiber. Okay. Now this is the class. Now come to subclass. So whether there is tetra axon spicules or monoaxon spicules. So we place them in subclass Monoxonida. Now come to order. Only there are large size of spicules are present and they are found in fresh water. Okay, and lots of spongine fiber. That's why we place the animal in Haplosclerina, order Haplosclerina, and genus Spongilla. Wherever you are writing genus, you must underline it. In this way, you can write the classification of the animal in that is given in museum specimen. Thank you everyone for watching us. You may like us, share our videos and share your views also, and subscribe the channel for getting update about our new videos. Thank you everyone.